Dear friends, I have discussed a topic stains and staining procedures in three different parts so far. In the first part, I have told you about necessity or importance of staining. I have also given definitions of different terms like stains, mordants and chromogens. Have seen about similarities and differences in stains and dyes, and at the end of the first part have seen the types or classification of stains as acidic stains, basic stains and neutral stains along with examples and their applications. In the second part have seen about types of staining procedures, where broadly have seen negative staining and positive staining. For positive, we have taken example of monochrome staining, and in this negative and monochrome staining we have discussed in detail about the procedures, the mechanism of staining and their applications. And in the last part, that is part 3, have seen about differential staining, where have seen that this is the staining procedure that will differentiate bacteria in different groups. And under this have seen two examples of gram staining and acid fast staining. In both the differential staining procedures have seen about the procedure, mechanism and applications of these staining procedures. Particularly in gram staining, we have explained the mechanism by considering different theories as lipid theory, peptidoglycan theory and magnesium ribonucleate theory. And in addition have also discussed about the mechanism of acid fast staining. And now we are going to discuss last part of the topic stains and staining procedures. Under this we are going to discuss special staining procedures. As the name itself indicates, these are the staining procedures that are used to demonstrate special cell structures or cell parts. This will include the demonstration of anatomical parts of bacterial cells, for example, cell wall, capsule, flagella and various constituents which are present in the cell cytoplasm, including nuclear material and the reserve food material. So initially we will discuss the definition. That special staining procedures are the procedures that are used to stain and demonstrate special cell structures, including the anatomical parts and cytoplasmic constituents. Under these procedures we are going to discuss three special stainings. First, staining of cell wall. Cell wall, as you are aware, is the outermost part of a bacterial cell that cannot be demonstrated by the staining procedures that we have described so far, and that's why we have to use a special staining procedure. And the method that is used to stain cell wall is Chance's method. This name is given in honor of scientist Chance, who devised this staining procedure for demonstration of cell wall. What is the feature of this staining? So far we have discussed that in a single staining procedure, maybe monochrome, maybe feature of this special staining for cell wall demonstration is that this staining procedure uses both acidic as well as basic stains. We have seen that in other staining procedures that we have discussed so far, there is use of either acidic or basic stains. But this procedure involves use of both acidic as well as basic stain. The acidic stain used in this staining procedure is Congo red, while the basic stain used is 0.5% new fuchsin. Now we will see what is the procedure for demonstration. Initially, you have to prepare a smear from the given suspension on a clean grease-free slide. Smear is to be air dried and heat fixed. After heat fixation, the smear is flooded with 0.5% new fuchsin, a basic stain, and it is kept for about 1 to 2 minutes. Then slide is washed with water. Then finally smear is covered with an acidic stain called 0.5% Kangorade and allowed to react for 5 minutes with smear. After that, slide is washed with water, slide is air dried, have to put a drop of oil on smear, and the slide is to be observed under oil immersion lens. Now, what are the observations? You will observe that cell wall is dark pink in color, while the cell cytoplasm is either colorless or faint pink in color. Now we will observe that why this wall is dark and cytoplasm is faint pink or colorless. So mechanism of staining states that new fuchsin stain, a basic stain that is applied initially, stains both cell wall as well as cytoplasm. But when 0.5% Kangorade, an acidic stain, is applied in the latter stage of staining, it selectively removes the color from cell cytoplasm, while the color in cell wall is not removed by Kangorade, mainly because it is more acidic and it accommodates more number of new fuchsin molecules. As a result, the color from cell wall is not removed, but cytoplasm becomes colorless, and in this way you will get a clear contrast between the cytoplasm and cell wall. It is the application of special staining. Now this cell wall staining normally of course is helpful for demonstration of bacterial cell wall. Now this is the figure that will show how the observation is done. You will find that these are rod shaped bacteria and it will have a cell cytoplasm which is colorless, that is shown by arrow, while the cell wall it becomes pink in color. Another special staining procedure that we are going to discuss is capsule staining. Capsule, it is present in some of the bacteria as an additional structure outside the cell wall. Now it has about 98% of water, so it is full of water, and for its demonstration we have to use a special staining which is known as Manéval's method. Again, the name is in honor of Manéval, who designed this particular staining for demonstration of capsule. We will see what are the features of capsule staining. Now capsule staining, like cell wall staining, uses two stains, of which one stain is a basic stain that is called Manéval stain, while the another stain is Kangorade, that is an acidic stain. The Manéval stain, it contains the actual dye, that is acid fuchsin, which of course is a basic stain, and in addition it has a mordant called ferric chloride. While the acidic stain is Kangorade, but the Kangorade used in capsule staining differs from cell wall in concentration. In cell wall staining there is use of 1% of Kangorade. In cell wall staining there is use of 0.5% of Kangorade, but in Manéval's method of capsule staining there is use of 1% Kangorade. Now the capsule staining, it uses the principle of negative staining, will mean that in this staining procedure background is colored like negative staining, and that's why it is supposed to be using a principle of negative staining. And the third feature of this capsule staining is that there is no heat fixation step, because when we will apply heat, the capsule will undergo dehydration because it contains about 98% of water, and as a result it will shrink and it will not be possible to visualize it clearly. We will see the procedure that is followed for capsule staining. The procedure is a bit different. In rest of the staining procedures that we have discussed so far, the smear is prepared within the marked area that is prepared on the slide. But in this staining procedure, like negative staining, the smear is prepared on complete slide surface. So for that, you have to place a drop of bacterial suspension on one end of a clean grease-free slide. Near to the drop of bacterial suspension, you have to place a drop of 1% Kangorate. Then both the drops, they are mixed properly, and with the help of another clean slide, you have to make a smear on complete slide surface. Once the smear is prepared on complete slide surface, smear is air dried, but heat fixation is completely avoided in the staining procedure. Once there is complete drying of the smear, then smear, that is total slide surface, is covered with a manual stain and it is allowed to react for 2 minutes. After 2 minutes, the excess of the stain is discarded and the smear is air dried. A drop of oil is placed on the smear and the smear is observed under oil immersion lens of the microscope. We'll see what is the observation. We'll find that the capsule that we are going to demonstrate is colorless. Cells, they are red in color while the background is blue in color. You will see why this type of different colors they are observed. That we are going to discuss the mechanism. That the procedure uses a basic stain, that is acid fuchsin, and acid fuchsin, it colors the bacterial cells in presence of a mordant called ferric chloride. And the Kangorate, it colors the background because it's a acidic stain. The capsule is remaining colorless. The reason is that the capsules, they are non-ionic in nature, meaning they do not have any electrical charge, and hence they do not have affinity for either acidic or a basic stain, and that's why the capsule, it remains colorless. Let's see what is the application of this staining. That this staining procedure is used to demonstrate the capsules in capsulated bacteria. Diagrammatic presentation of capsule staining, and in this diagram we will observe that the capsule is colorless, cells, they are red or pink in color, while the background is blue in color. Now color of the background, blue, it may be red, and red colored background is observed usually when we wash the slide after application of Kangorate. And finally we are going to discuss a third example of special staining called metachromatic granule staining. Now these are the granules which are working as reserve food and they are present in cell cytoplasm. Now metachromatic granules, they are called as metachromatic because they show a metachromatic effect. Metachromatic, it's a Greek word where meta means change and chroma means color. This will mean that these granules, they appear of a different color than the color of applied stain. Normally, so far we have seen so many staining procedures in which the objects, they appear of a particular color, and that color is a color of applied stain. For example, when we apply methylene blue, cells are blue in color. When we apply crystal violet, cells are violet in color. But in this staining procedure, especially metachromatic granules, they appear of a different color than the color of applied stain, and because of this is the granules, they are known as metachromatic granules, and the effect is called as metachromatic effect. In this particular staining, when we apply blue color, the granules, they appear black. So this is known as metachromatic effect. In addition to this name, the granules are also known as Babes Ernst granules after their inventors, that is Babes and Ernst. The granules also are known as volutin granules because they were discovered for the first time in a spiral shaped organism called Spirillum volutans. The main feature of these granules is that they are serving as a reserve food, and especially they are polyphosphate granules. Ultimately, they will provide phosphorus in adverse condition where in the environment phosphorus is not available, and that's why it is known as reserve food. So these are known as intracellular inorganic phosphate reserves. We will see what is the feature of this staining. The staining procedure involves use of Albert stain, and there is also use of a mordant known as Albert's iodine. The feature of Albert stain is that it contains two dyes, that is toluidine blue and malachite green. So it is a mixture of two basic stains. Mordant, that is Albert's iodine, like Gram staining, this also is reacting with Albert stain and helps the stain molecules to the attachment of to the bacterial cells. You will see what is the procedure. That you have to prepare a smear like other ordinary staining procedures. Usually smear is prepared from a suspension. Normally a curd suspension is used because the bacteria which are present in the curd, called lactobacillus, they have a reserve of metachromatic granules. So from a curd suspension you have to make a smear on a clean grease free slide. Smear is air dried and heat fixed. After heat fixation, the smear is treated with chloroform for 30 seconds or xylene for 1 minute. Purpose of this particular step is to remove the fats that are present surrounding the cells. If these fats they are not removed, which are present around the cells, ultimately stains cannot reach to the cells and staining is not possible. After defatting with xylene or chloroform, then slide is washed with water, and after that smear is flooded with a first stain called Albert stain, which is allowed to react for about 5 minutes. After 5 minutes, the Albert stain is removed, it is discarded, and smear is covered with Albert's iodine for 1 minute. After 1 minute of the mordant treatment, then slide is washed with water, air dried and observed under oil immersion lens of the microscope. What are the observations? That the cells they are green in color, while the volutin granules they are blue black in color. We will see what is the mechanism of this staining. As already I cleared that Albert stain contains two basic dyes, toluidine blue and malachite green. While the granules they are highly acidic because these are polyphosphate granules and we know that they have negative charge over them. So these granules they are highly acidic and hence are stained by a acidified basic stain. Especially the toluidine blue, it will color the volutin granules, but because of the metachromatic effect, the granules they do not appear blue but they appear black in color. Then pH of the Albert stain is about 2.8. So this pH, it is acidic to cell cytoplasm while it is basic to granules, and that's why the granules they are stained by basic dyes. Toluidine blue preferentially colors the granules and hence granules appear blue in color. While the malachite green, it colors the cell cytoplasm. So cells they appear green in color. Albert's iodine, as we have seen, it's a mordant that will fix toluidine blue molecules by precipitation that will help in coloring of these granules. So this is the mechanism, and here we will find that the granules they are blue black while the cells they are green in color. You will see what is the application. This particular staining procedure is helpful for diagnosis of a disease known as diphtheria. We call this in Marathi as Ghatasarp. It's a causative agent is Corynebacterium diphtheriae. It's a respiratory tract disease, and these organisms they have ability to deposit metachromatic granules. When a sample from a patient is used for making a smear, and when we will subject this smear to this particular staining by Albert's method, you will find that the cells will show black granules, which will be the indication of presence of these organisms and ultimately the disease of the individual from which the sample has been collected. Now this is the diagrammatic presentation where you will find that volutin granules they appear blue black in color. The stain used is toluidine blue, but because of metachromatic effect they are showing different color. While the cells they are green in color. Cells will mean cytoplasm of the cells will be green in color. So in this way, metachromatic granule staining will be helpful for diagnosis of a disease known as diphtheria. So this is the end of this particular chapter of stains and staining procedures, which we have discussed in 4 different parts, and that is the completion of the topic stains and staining procedures. Thank you.